Yes, body cells can lose their identity over time, and this process can occur in several ways. Here's how it can happen. Number 1, cellular aging, senescence. As cells age, they accumulate damage to their DNA, proteins, and other cellular components. Over time, these changes can alter a cell's identity, leading to a loss of function. Senescent cells often lose their ability to divide and may no longer perform their specialized functions effectively. They can also start to produce inflammatory signals, contributing to tissue dysfunction. 2. Epigenetic changes. Cells maintain their identity through specific patterns of gene expression, regulated by epigenetic markers like DNA methylation and histone modification. Over time, these epigenetic marks can change due to environmental factors, aging, or other stresses, leading to a loss of the original identity of the cell. This can result in cells behaving differently than they should, potentially leading to diseases like cancer. 3. Stem cell exhaustion. Stem cells are responsible for renewing tissues by producing new specialized cells. As these cells age or are exposed to environmental stress, they can lose their potency or ability to differentiate into specific cell types. This loss of regenerative ability can cause tissues to degrade or malfunction, contributing to aging and the loss of cellular identity. 4. Chronic inflammation. Prolonged inflammation can alter the microenvironment of tissues, leading to changes in how cells behave. Chronic inflammation can push cells towards a less specialized, more generic state, or even cause them to adopt harmful identities, such as those seen in cancer. Cancerous transformation. Cancer can be viewed as an extreme loss of cellular identity. Normal cells undergo mutations that disrupt the controls on their growth and function, leading them to behave in ways that are completely at odds with their original purpose. Cancer cells often lose their specialized functions and instead focus on uncontrolled growth and division, spreading through the body. 6. Mitochondrial dysfunction. Mitochondria are the powerhouses of the cell and play a key role in maintaining cellular health. As they accumulate damage with age, their reduced efficiency can lead to energy deficits and the disruption of normal cellular functions, contributing to a loss of identity. 7. Telomere shortening. Telomeres are protective caps on the ends of chromosomes that shorten each time a cell divides. When they become too short, it triggers a state called replicative senescence, where the cell can no longer divide and may lose its functional identity. 8. Loss of tissue-specific signals. Cells maintain their identity in part through signals from their environment. If these signals diminish due to aging or disease, cells might lose their specific roles and functions, contributing to tissue degeneration. These processes are a normal part of aging, but can also contribute to diseases, highlighting the importance of maintaining cellular health to preserve the function and identity of cells over time. Slowing down or stopping the loss of cellular identity and the processes associated with aging is a major focus of research in biology and medicine. While we can't completely stop these processes, several strategies have been identified that may slow them down. Step 1. Healthy lifestyle choices. Diet. A balanced diet rich in antioxidants, vitamins, and minerals can help reduce oxidative stress, which is a key factor in cellular aging. Diets like the Mediterranean diet, rich in fruits, vegetables, nuts, and healthy fats, are associated with better cellular health. Exercise. Regular physical activity has been shown to improve mitochondrial function, reduce inflammation, and enhance DNA repair mechanisms. Exercise also helps maintain muscle mass and function, which are important for overall cellular health. Sleep. Adequate and high-quality sleep is crucial for the repair of DNA and the maintenance of healthy cells. Sleep helps in clearing out toxins from the brain and other tissues, supporting overall cellular health. 2. Reducing chronic inflammation. Anti-inflammatory diet. Consuming foods that reduce inflammation, like omega-3 fatty acids, found in fish, flaxseed, and walnuts, turmeric, and green tea, can help maintain cellular health. Stress management. Chronic stress contributes to inflammation. Practices like meditation, yoga, and mindfulness can help reduce stress and its impact on cellular aging. 3. Protecting telomeres. Telomerase activation. Some research suggests that activating telomerase, the enzyme that can extend telomeres, might slow aging. While telomerase activating therapies are still experimental, lifestyle factors like stress reduction, regular exercise, and a healthy diet have been associated with longer telomeres. Supplements. Some studies suggest that supplements like omega-3 fatty acids and certain vitamins, e.g., vitamin D, may help protect telomeres, though this is still an area of ongoing research. Matter 4. Caloric restriction and fasting. Intermittent fasting. Intermittent fasting or caloric restriction has been shown in animal studies to extend lifespan and improve cellular health by reducing oxidative stress and enhancing DNA repair mechanisms. Mimicking caloric restriction, certain compounds like resveratrol, found in red wine, and metformin, a drug used to treat diabetes, are being studied for their ability to mimic the effects of caloric restriction, potentially slowing aging processes. Matter 5. Regenerative medicine. Stem cell therapies aim to replace damaged or aging cells with new, healthy ones. Although this is still in the experimental stages, it holds promise for reversing some aspects of cellular aging and restoring function. 6. Epigenetic interventions. Epigenetic drugs. Researchers are exploring drugs that can modify epigenetic markers, potentially reversing age-related changes in gene expression. While this is a developing field, early results are promising in animal models. Lifestyle factors, diet, exercise, and stress management can also influence epigenetic markers, potentially slowing the loss of cellular identity. 7. Antioxidant, supplements and diet. Antioxidants help neutralize free radicals, which can damage cells and contribute to aging. Consuming foods high in antioxidants, like berries, nuts, and dark chocolate, or taking supplements such as vitamin C, E, and coenzyme Q10 might help protect cellular integrity. 8. Senolytics, clearing senescent cells. Senolytics are a class of drugs that selectively target and eliminate senescent cells, which are cells that have stopped dividing but haven't died. These cells can contribute to aging and inflammation. Research in this area is ongoing, but it shows potential for reducing the impact of cellular aging. 9. Genetic interventions. Gene therapy aimed at repairing or enhancing specific genes involved in aging and cellular identity is a cutting-edge area of research. Although not yet widely available, it represents a future direction for slowing down aging. 10. Regular health monitoring. Medical checkups. Regular health screenings and monitoring for age-related diseases, like cardiovascular disease, diabetes, and cancer, can help detect problems early and prevent or slow their progression. While aging is a natural process, these strategies can help maintain cellular health, potentially slowing down the loss of cellular identity and the effects of aging. Ongoing research continues to explore new ways to enhance and extend cellular function. Glucose spikes, or rapid increases in blood sugar levels, can have several detrimental effects on cellular health, contributing to the cellular changes associated with aging and disease. Here's how glucose spikes impact cells. 1. Oxidative stress. Reactive oxygen species, ROS. High glucose levels can lead to the production of reactive oxygen species, ROS, which are highly reactive molecules that can damage cellular components, including DNA, proteins, and lipids. This oxidative stress accelerates cellular aging and can contribute to the loss of cellular identity. Mitochondrial damage. Excess glucose can impair mitochondrial function, leading to inefficient energy production and increased ROS generation. Mitochondrial dysfunction is a key factor in aging and many chronic diseases. 2. Glycation. Advanced glycation end products, AGEs. When glucose levels are consistently high, sugars can bind to proteins, lipids, and nucleic acids in a process called glycation. This leads to the formation of AGEs, which can disrupt normal cellular function by altering protein structure and function. Tissue damage. AGEs accumulate in tissues over time, contributing to stiffness, reduced elasticity, and dysfunction in organs such as the skin, blood vessels, and kidneys. They are also implicated in the development of age-related diseases like diabetes and Alzheimer's. 3. Inflammation. Chronic inflammation. Repeated glucose spikes can trigger an inflammatory response, leading to chronic low-grade inflammation. This inflammation can damage tissues, contribute to the loss of cellular identity, and promote aging-related diseases. Cytokine production. High glucose levels can stimulate the release of pro-inflammatory cytokines, signaling molecules that exacerbate inflammation and contribute to cellular stress. How to 4. Insulin resistance. Cellular stress. Frequent glucose spikes can lead to insulin resistance, a condition where cells become less responsive to insulin, the hormone that regulates blood sugar levels. Insulin resistance forces the body to produce more insulin, which can stress pancreatic cells and lead to metabolic dysfunction. Metabolic syndrome. Insulin resistance is a major component of metabolic syndrome, which is associated with increased risks of cardiovascular disease, diabetes, and accelerated aging. Now to 5. DNA damage. Genomic instability. High glucose levels can cause DNA damage either directly through oxidative stress or indirectly by promoting inflammation. This damage can lead to mutations, loss of cellular function, and increased risk of cancer. Telomere shortening. Chronic high blood sugar has been linked to accelerated shortening of telomeres, the protective caps on the ends of chromosomes. Shortened telomeres are associated with cellular aging and the loss of cellular identity. Number 6. Disruption of cellular signaling. Impaired signaling pathways. High glucose levels can disrupt normal cellular signaling pathways, including those involved in growth, metabolism, and survival. This can lead to abnormal cell behavior, such as uncontrolled growth or failure to respond to stress. Endothelial dysfunction. In blood vessels, high glucose can impair endothelial cell function, leading to atherosclerosis and other cardiovascular problems. Cellular senescence. Promoting senescence. Frequent glucose spikes can push cells into a state of senescence, where they stop dividing and lose their function. Senescent cells contribute to tissue dysfunction and aging by secreting pro-inflammatory factors and other molecules that damage neighboring cells. 8. Autophagy inhibition. Reduced cellular cleaning. Autophagy is a process by which cells clean out damaged components. High glucose levels can inhibit autophagy, leading to the accumulation of damaged proteins and organelles, further contributing to cellular aging and dysfunction. 9. Epigenetic changes. Altered gene expression. Chronic high glucose levels can lead to epigenetic modifications, such as changes in DNA methylation patterns that alter gene expression. These changes can contribute to the loss of cellular identity and increase the risk of age-related diseases. 10. Impact on stem cells. Stem cell exhaustion. High glucose levels can impair the function of stem cells, which are crucial for tissue regeneration. This can lead to the depletion of stem cells over time, reducing the body's ability to repair and renew tissues, thereby accelerating aging. Managing glucose spikes. To minimize these effects, it's important to manage blood sugar levels through diet. Eating a balanced diet with low glycemic index foods, reducing sugar intake, and consuming more fiber can help prevent glucose spikes. Regular exercise. Physical activity improves insulin sensitivity and helps regulate blood sugar levels. Monitoring. Regularly checking blood sugar levels, especially for individuals with diabetes or prediabetes, can help manage and prevent spikes. Medications. For those with diabetes, medications like metformin can help manage blood sugar levels and reduce the risk of spikes. By managing glucose levels effectively, it's possible to reduce the impact of glucose spikes on cellular health, thereby slowing down the processes associated with aging and disease.